Okay, thank you. So I am Anders Finstad. I am currently serving as a co-PI for the Living in Norway Ecological Data Network. I'm affiliated with the Norwegian University of Science and Technology, and I'm a professor with the Department of Natural History, where I also serve as a data curator. And my particular responsibility at the moment for the Living Norway Ecological Data Network is in data standardization. Hence the title and the theme of this talk. And there's a recurring question that comes up when I talk about data standards and ecology, and that's sort of, this is not possible. And my aim with this talk was to show, yes, it is possible. Hence, the talk title should really be sort of, can we standardize ecological data? And the subtitle would be, yes, we can. But then again, that drifted me into the slogan, make ecology great again. And I sort of, no, that's not selling in 2020. Let's hope not. So anyway, this comes with a bit of warning. First of all, to the geeks out there, this will not be a very technical-oriented talk. It also comes with a promise to the non-geeks out there that this will not be a very technical-oriented talk. But first of all, needs sort of, what are we talking about here? So, with the talking about data standards, what really is this? I like to say that it's sort of three different parts. It's a content descriptor, which is what is this about, and we commonly refer to that as metadata. It's about the data structure, which is sort of the technical aspects, what's the file formats, and etc. And it's also about information content and retrieval of that, which is a common set of terms to describe your data, what you call your columns, and what do they contain, etc., etc. Now, that being described, as you mentioned in the previous talk, there's something that's called fair data. Well, it's a fundamental principle that in order for data to be useful for reuse, they should be findable, they should be accessible, and they should be interoperable with other data. As Alan pointed out in his previous talk, they need to flow together in order to do joint analysis, for example. And of course, they need to be reusable. There's several sort of things that needs to be in place to make this function. And it's important to note in this respect that FAIR is not a data standard per se. However, in order to make data fair, they requires a set of underlying standards in order to make data talk together, in order to make search engines find your metadata, etc., etc. So all of these points here need to have various different standards, and for ecology we operate with a different set of substandards again. So some standards are very common and some are narrowing down depending on which field you are working in. This is my take on what you say. This is the cornerstones of the data life cycle, which doesn't add up. But the data life cycle is sort of, we talk about data as going first being reusable, hence it's a cycle. Well, first it's the planning and collecting of it, which is the empirical part of it. Then it's the private things done by individual researchers, which is quality check, analysis, and storage. And then comes the interesting part, which is sort of new for the whole concept of open science and open data, which is you need to describe your data, you need to archive them, you need to share them with the world, and you also need to rediscover and reuse them. So it is often, the first part has been done by researchers repeatedly, but it's the last part here. We do the quality check, analysis, and storage, and then describe and share data. That's new to most of researchers in our field, and hence that's a lot of questions. What kind of standards should we use there? How should I do this? And it's also coming up when reusing data. How should I reuse data in a sensible way and an open, reproducible way? This is sort of what we're gonna try to tackle for ecology in the Living Norway network. Much of these issues are related to the perception of ecological data as very heterogeneous. I mean, it's a range of sampling techniques, a range of methods, and how could you possibly make these flow together? It's a problem that's been noted in the literature also previously, as this is a paper from 2019. And my claim here is that ecological data is not really different or disparate. They are just different traditions for how you express your data from different subfields of ecology. Either if it's coding your individuals in columns or in rows, or if it's raster data, or if you have them in a very highly normalized database, etc., etc. My claim to this is that there actually are common denominators for ecological data that make this very possible of sewing them together in a standardized way and sharing them together. So this I call a conceptual schema, or it refers back to what you call a data schema, but it's really a conceptual data model for how ecological data are built together. And I mean, everything we do for field ecological data and now sort of sample out in nature has a location. It might be a lake or an area, it might be a vegetation plot or whatever, but it has a location that can be georeferenced. At that place, there is something happening. An event, a sampling event, where someone goes out and observes something. It might be with a big research vessel, here illustrated by NTNU research vessel Gunneris, or it might be a very small research vessel, here by the NINA research vessel, MS Edna. Anyhow, it might be by a camera trap or automatic or remotely by radio telemetry. Doesn't matter, you do something there. Then you do an observation, which you term an occurrence. And it might be a batch of organisms, it might be a single organism, it might be evidence of an organism being there from a DNA or other trace, or it might be a picture from an automatic collection, or it might be some repeated measures of a radio-tagged individual. And you might do a sample of that again and bring back either whole specimens, part of a specimens, or specimens altogether. The point is that this is also different locations can be nested within each other. I mean, and events can be nested into each other, like, for example, the repeated samplings of the same plot. And then occurrences might have, there might be different scale you're observing on. It might be just nominal presence-only data, it might be binomial data, it might be an ordinary scale or true quantitative data, depending on. And at each of these points, you might have some additional measurements or facts of observations, either if the location has some names or whatever, or something about the event or of the occurrences. So this common set of describing data leads to the current practice where there are a number of ecological data sets shared today through data infrastructures, making them available and discoverable in a fair manner. And it's, in other words, it is possible to do it. One of the reasons, or this is possible, is that you have some data standards to build on. And this is where I get slightly technical and mention the most important data standard we had for this purpose, which is Darwin Core. And Darwin Core is a body of standards, in a broad definition, is a body of standards that are used and to facilitate biodiversity data sharing. In a very narrow definition, it's a library of a common set of terms that can be used to describe biodiversity data. It's maintained by the Biodiversity Information Standard Working Group, which is an international organization, and which is shortened to TDWG, or shortened TADWG. In the histories behind this is that this is the earlier taxonomic something working group. Now, how does this look? Simply a common number of terms. How would I describe an occurrence, for example? Well, you have a set of terms that refers to giving the observation and unique ID and a name of the organism, and you might have some measurements of quantity, for example, biomass, and it's etc., etc. Fairly simple example, but building up on this common body of standards, you're able to actually publish and disseminate quite complex data types. So that being said, I am going to go through with you a few examples of what kind of data that actually are shared through the Global Biodiversity Information Facility today. Now, Dag Andresen going to come up next and talk more about DBIF, so I'm not going to give a brief more introduction to that, but just showing off that we have species inventory type of data, here exemplified by bumblebees and butterflies in Norway dataset from Nina. You might have panel data. Panel data is what we talk about when we say that this is repeated measures of a given individual throughout time, typically telemetry type of data. And you might have vegetation plots, like squares with a semi-quantitative abundance range. Or you might have line transects, either of the aquatic or terrestrial types. And of course, you might also have the traditional quantitative monitoring of animals or plants. Now, you might have less traditional methods, like eDNA-based inventories. And you might have camera traps, automatic detections of animals. Now, the main issues with this, or challenges, is yes, it is possible. We can share this type of data through Darwin Core as a standard, through the Darwin Core archive as a technical standard, and through ecological metadata language as a metadata standard for describing the data sets. However, a lot of these data are use Darwin Core in a slightly different way. So there's a heterogeneity in the application of how Darwin Core are used to code the ecological data. Just a brief example of 4 different randomly selected inventory type of data sets from DBIF, which all uses different types, uses Darwin Core in a slightly different way, meaning that they are not directly interoperable. The solution to this is, as we see it, that we need more community-derived recommendations, and possibly also more than recommendations from some of the funding agencies in order to how to publish and disseminate ecological data. This is an example that we've been working now through GBIF on how to publish sequence-derived data, meaning there's another word for eDNA-based data, and how to do that in a common standard format. Now, however, one of our main issues is, of course, still data stuck in the drawer. The lack of time, of resources, or of motivation or know-how of the individual researcher or organization to actually disseminate your data in a fair way is maybe the most significant hurdle we have. So our take on this is, how do we move forward? 3 short bullet points that we would like to address. We would like to build on existing standards, which is Darwin Core and the ecological metadata language as a metadata descriptor, and develop and employ community-driven best practices for verification of publishing and dissemination of ecological data. Second to that is to build on the existing infrastructure like particular DBIF, to facilitate and develop user-friendly tools for easing the technical barrier for standardization of data and also reuse of data. And I guess Matt is going to touch on this on his talk coming up later in the session. And also, last but not least important, is to increase the community know-how of sharing and publish and how to reproducible and transparently reuse ecological data. That needs to target both existing researchers, both our new upcoming researchers and students, and also to a certain degree our funding agencies and managers. And with that, thank you.
So we do have 3 minutes for questions, if you're willing to take any.
Do we only have one mic?
No, there is one there that one of you can hold.
That's better. Like this? Yeah.
Yep.
Okay.
So there's a question from Chloe. To what degree do you think that we might profit or not from also standardizing data collection protocols, methods, definitions, etc.?
Absolutely. We could profit quite a lot for that to some degree, where it comes to some type of typical management driven sampling protocols for water resource management, etc., etc. It's already standardized. I think it's overly optimistic to believe that researchers or the basic research community is going to really standardize their sampling protocols, because this is going into details of their experimental analysis, etc., etc. Don't think it necessarily hinders reuse and interoperability and integration of data that the sampling protocols slightly differ, as long as it's transparently coded and disseminated in the data. Yeah, hope that was sort of an answer.
Yep. I think we have time for one more, maybe. Yeah.
Yep.
Question from Rainer. How does, for example, indigenous and local knowledge fit into these schemes and in your approach? This kind of data is crucial for moving forward in biodiversity related issues, and the knowledge of local and indigenous people must not be ignored.
Well, I guess it falls into the scheme as far as it's observed. I mean, depending on what's the observation unit here, that's kind of different. It might be the people's perception of nature you observe and code as observing, and their territory as your location, or it might be their observations that's coded directly. That's kind of, I have no opinion on that particular, but in general, I think it fits, yes.
Thank you.